So first, I invite Xue Zong from Georgia Tech, and it will come. And her talk will be F.A.D.E, Fully Agentic Drug Engine, a conversational artificial intelligence platform for drug discovery.
Hi, everyone. I'm Xue. It's a great pleasure for me to be here today representing my team and talk about our project on F.A.D.E, which is a conversational AI workflow for accelerating drug discovery. So drug discovery is always known to be a lengthy and resource-intensive process. It typically requires 10 to 15 years and costs more than $2 billion for a successful drug candidate to enter the market. So to address these challenges, computational approaches have been very critical for the early stage drug development. However, most of the methods we have today are very task-specific and require human execution by domain experts. So since the drug development is inherently a multi-step and interconnected process, we think this fragmented implementation limits both the scalability and the efficiency of the current pipeline. So to address this limitation, here we propose an LLM-powered multi-agent workflow, which can automate and streamline the key tasks across the early-stage computational drug discovery pipeline. So by combining the LLM reasoning with the domain-specific tools, our workflow can perform data retrieval, molecule generation, property prediction, and also the binding affinity prediction. Here we summarize our overall workflow at the top of the slide. So first, the user describes their target of interest in natural languages. For example, you can ask our agent, can you suggest me some small molecule candidates that could target the EGFR target? With this input query, our agent will first extract the target information and the user requirements from this message. Then it will start searching the databases in a hierarchical order. If a co-crystallized protein ligand structure can be found from the RCSB database, our agent will directly download the structure with the highest resolution and separate the bound ligand from the protein structure. If only the apo protein structure is found, the agent will send it to F.A.D.E, which can be used to detect all potential ligand binding pockets. If none of the protein structure is available, our agent will find the sequence from the UniProt database, use both two to predict the 3D structure of the protein, and then send it to F.A.D.E for binding pocket detection. And after this step, we will send our input structures to our molecule generation agent, which will execute the deep SBDD package for de novo molecule generation. So in this step, we will evaluate the drug likeness of our generated molecules by our defined criteria, such as the empirical rule of 5, the QED scores, and the synthetic accessibility scores. The users can also customize their own success criteria and ask the agent to keep generating molecules until it meets the criteria. And after this, for the final step, we will send all the generated molecules to our agent so it can rank them based on the scores. And it will select the top 10 molecules, all of them, and send them to both to predict the 3D structures and also the binding affinities. So we have tested our workflow on 2 case studies, but due to the time limit today, I will only show the results for the EGFR case at the bottom. So in this case, our agent successfully retrieved the protein ligand structure, co-crystallized structure from the RCSB, and separated the bound ligand as the reference ligand. So at the bottom left is the simple 2D visualization of our generated chemical space. The red point on the right is the reference ligand. So when we, in our molecule generation agent, we also set up a Tanimoto similarities threshold. So we ask the agent to generate molecules that are different from the reference ligand if there's a no ligand available. And after that, the 2 structures highlighted are the top ligands we find, and we use both two to predict their structures and also predict their binding affinities. So given the binding affinities we predicted and the comparison to the reference ligand, we think these 2 ligands are potentially moderate to strong binders that are worth further exploring. So in summary, our workflow is one step further in applying the AI agents for accelerating the drug discovery process, and it can shorten the turnaround time to generate some new ideas to be tested out. And the modular architecture also allows the integration of new tools and models as it comes up. So ensuring. Oh, okay, sorry.
So, hi everyone. Today I will to introduce about our project, the PostaNet, a deep learning-based protein thermostability predictors. The PostaNet will require the 3D structure of the proteins as the input, and it will to predict the protein thermostability change on the point mutations, which is to input by the users. The model will predict if the point mutations increase or to decrease the protein thermostability. Here, we have to use the geometric vector perceptions, the graph neural network, as the core of our framework. The GVP and the GAN can learn the scalar and the vector values over the 3D Euclidean space, which have to show the potentials in predicting the protein properties. So for the database, we have to collect the protein thermostability change on the point mutations data from all the established database and to combine the data together to form the first comprehensive protein thermodynamic database called the PostaDB. And this database contains the both single point and the multiple points mutations. Because previously the multiple point mutations data didn't have the testing set, so we introduced the novel clustering methods which to split the multiple point mutation data into the training and the testing set. And at the same time, the method has to ensure the generalization of the models. To solve the problems of the data limitations, we have to develop the novel data augmentation methods called the thermodynamic loop, and to combine with the anti-symmetric augmentation methods to enlarge the data size. With the enlarged data sets, we train our models. We have to use the testing set to evaluate how's the performance of the PostaNet, because originally the testing set contains the large amounts of the destabilizing mutations. In order to ensure our PostaNet can to predict accurately for both the destabilizing and the stabilizing mutations, we have to introduce the reverse mutations for the testing set. Here, we call the protein A mutate to the protein B as the direct mutations. And if the protein B mutate to the protein A, we will to call it as the reverse mutation. The reverse mutation's thermostability change is the opposite of the direct mutations. So for the originally of the testing set, it is called the direct mutations. So we generate the reverse mutations for the testing set. So as the performance, we can see the PostaNet have to show the high and accurate performance in both the direct and the reverse mutations, which have to show the less bias and to better performance compared to the state-of-art methods. To further to validate the performance of the PostaNet, we use the PostaNet to predict the thermostability change of the several single point and the multiple points mutations for the humanized nanobody J3. And after the experimental validations, 4 out of the 5 single point mutations and all the multiple point mutations were predicted correctly by the PostaNet. And the PostaNet also predicted correctly for the 2 positive mutations. And both of them have to show it can improve the thermostability of the HUJ3. On the summary, the PostaNet have to show the potentials to accurately to predict the protein thermostability change. Thank you.
Good afternoon, everyone. My name is Xueyang. I don't have fancy models, but I wanted to talk about the new design for biological discovery, one that bridges the gap between computation and the wet lab. In my opinion, with the introduction of many great foundation models, a powerful engine is already built for us. So this changes our focus from model to a more holistic experimental design. This is our lab's version of human-in-the-loop cycle, where we first frame the question precisely, and then we fine-tune a pre-trained model and interpret the model, and finally validate those discovery back in the lab. So now let me give a more concrete example. It all begins with framing the question. In another word, This means defining a biological question that's specific, and the scope of question has to match the complexity of your data. For example, here we have data from RNA sequencing experiments on cell treated with PRMT5 inhibitors. PRMT5 inhibitors, which are promising cancer drugs that's in clinical trials. And using splicing analysis of this cell line, we find we identified about 3,500 of detained intron, and half of them were upregulated in red here, and half were not. This gives naturally two classes and allow us to frame a precise question. Within 150 nucleotide of the 5' splice site, are there any regulatory sequence that determine if the intron will be upregulated by the drug? So next, we move to the model. Since we have labeled DNA sequence data, we choose to fine-tune a genomic model, DNA BERT, and the next critical step is interpretation. So we use two methods, backpropagation-based method and in silico mutagenesis. So to basically find what the model actually learned. Let's first look at the known fact before the model. This is a simple overlay of the data, which is nucleoside frequency plot. You see the signature GT at the splice site, which is the signature of this, which is what we expected. And you also see in class 0, there's overrepresentation of the Gs at -1 and +5 position. So now, let's look at what the model learned. This logo shows the feature importance from the model for individual instance and for a whole class. So basically, what's the first thing you see? The GT is gone, right? This is perfect. Because GT is in most of the instances in both classes, it's not a differentiating feature. So the model learns to ignore that. And what did it learn? We do still see the 2G, -1 and +5, which is great. What's more, there are more new discoveries. Let's look at this example here. Although the sequence has 2G, it's assigning to class 1. Why? The interpretation showed us the 2G did contribute negatively, but the other sequences near in this intron have a stronger positive influence that push it towards class 1. And this is the type of nuances we can only get from an interpretable model. Okay, hopefully I've convinced you how powerful this model interpretation can be. And what's even more important is that these are not just computational patterns, they're easily testable by a splicing reporter. And last, I want to emphasize that this discovery is not the end. This is actually a launchpad for many new exciting ideas and questions. For example, we can go through all of the sequences, features to find new regulatory motifs and ask, is it a common pattern? What protein binds here? And perhaps more interestingly, we can also cross-reference features with known human SNPs to potentially infer patients' drug discovery in clinical trial. In conclusion, foundation models are changing how we can do biology here now. And in many cases, bottleneck is not computation, it's efficient design. And this framework isn't meant to replace rigorous experiments, but to expedite them. Thank you very much for having me.
Thank you so much. And finally, Imran Haque from Kimia Therapeutics.
This is my reward for bragging last night at the speaker's dinner that I could give PowerPoint roulette. There we go. All right. Hey all, I'm Imran Haque. I'm CTO at Kimia Therapeutics. We are a chemistry platform startup based in Berkeley, focused on radically transforming the scale of what can be achieved in hit to lead, as well as chemistry model development. The way that we do that is by integrating synthesis of molecules at nanogram, nanoliter scale with screening on those same molecules in an integrated platform, with machine learning driven integration of thousands of data points that we deliver per hit to lead cycle to deliver a dynamic exploration of chemical diversity. And each of those words really matters there. This is informed by my past experience, for the last 5.5 years, I was at Recursion Pharmaceuticals, where we generated an enormous amount of data. But one of the challenges that we ran into is that although biology operates at a huge scale, chemistry operates a lot more slowly. And so if you look at the progress of a standard chemistry program, folks will identify hits. You may be able to do 1, maybe 2 rounds of catalog expansion of those hits from commercially available molecules for, call it, $100 a molecule in a few weeks. But very rapidly, you end up having to do custom synthesis where each molecule costs you $1,000 and you'll get your results next quarter. That's really challenging, not just for chemists in hit to lead, but I think really challenging for the folks in this room trying to do machine learning, because now you're trying to build models off like 10 molecules per cycle. So how do we address this? We address this by dealing with this on the data side. This starts at the top left. We're doing chemical synthesis at the nanoscale, where we're making 1,000 to 100,000 molecules per cycle at nanogram scale. So already you're putting 2 to 4 orders of magnitude in the chemical breadth that you can explore per cycle. This is then screened in direct-to-biology and direct-to-ADME format, where we run these in functional, biochemical, and cellular screens. So rather than looking at purely biophysical data where you might see that there's an interaction, but you don't know whether or not it's relevant or functional in the system you care about, you're actually getting information about the function of the molecules that you make. We then take interesting molecules. Because we're operating at such scale, we can afford to say, maybe 90% of the molecules aren't functional, but the 10% that are, we can then zoom in, synthesize and purify them at the microscale at hundreds of molecules per week, and then integrate this into synthesis-aware machine learning capabilities to integrate reactivity, potency, and selectivity data for the next round. On my poster, you'll see examples of this, where we apply this for everything from de novo hit finding, diversification of molecules and molecular scaffolds guided by potency, multi-parameter optimization, as well as using this data to drive foundation model development. So what I'll do in the next couple of minutes is just walk you through one demonstration of this in how we dealt with a very challenging selectivity challenge. I think Tom's presentation in the morning presented a really great example where he talked about needing to drive the selectivity for their molecule IM1363 versus EGFR in order to prevent dose-limiting toxicities. This is a really broad problem across a lot of targets in oncology and beyond, where there's a wealth of targets that have lots of genetic evidence, that have lots of validation that they would be great. The problem is that there are very closely related family members that you have to be selective against in order to actually be an effective therapeutic. And these present really, really difficult chemistry challenges. So we took one of these challenges on. This was an example where there's a target that has greater than 88% identity, residue identity in the binding pocket against 2 other off targets. We started with a non-selective tool molecule up here in the middle. You can see that it's actually stronger on these anti-targets than it was on the target. After 2 cycles of optimization on the Atlas platform, so again, this is synthesis, screening, purification, machine learning guided design. In this case, using models that are optimizing both potency on the target as well as selectivity, anti-potency on these other 2 targets. We're able to increase the selectivity by a factor of 250-fold. And we got a molecule that was 34 nanomolar against the target and up in the micromolar range against the anti-targets. So I think there's a really exciting platform to generate exploration diversity as well as data for model building. Come see me at poster 7 during the break. Thank you.
All right, I heard the applaud from outside. So I think we already gave a round of applause to all our speakers. But I would like to welcome all our 4 lightning talk speakers for 1 or 2 questions, if anyone has. Please come here, and we'll see if there is any question before we take the lunch break.
I have a question for Xue.
Yes.
So you've now predicted binding affinities or ranking molecules. How do you plan to validate your predictions?
That's a very good question. We don't have the collaboration with the experimental chemists to validate that ideas, but we hope this could work in a more collaborative setting so people can take our ideas to see if it could be synthesized really experimentally, or we can do a similarity comparison to any known drug candidates from the literature to see if that's already being explored in a similar way.
Yeah, I think that's going to be really critical to validate
Yes.
your method. Second question is, so you showed an example, EGFR, where there's a lot of structural information available
Yes.
for that target.
Yes.
And you mentioned your pipeline that if there is no structural information, you would use bolts to predict a structure.
Yes.
Have you done any kind of comparison to show, like, kind of what Woody was pointing to earlier, the correlation or your ability to predict novel, or tightly binding molecules if there isn't a lot of structural information? Is there that sort of linear correlation that?
So we haven't done that studies by ourselves. For the other case study we have tested is when we have the APO structure, how can we identify the multiple binding pockets from that and screen different ligands, generate different ligands for different pockets. But that's a really good direction we should take into consideration.
Thank you.
Thank you.
All right, so we will give one more round of applause to all our speakers this morning.